Welcome student to our session 13 about research sampling. By the end of this session, I'm expected you to be able to define the common terms used in sampling, to be able to identify populations to be studied, to be able to describe common methods of sampling, to be able to explain reason for sampling, and you should be able to describe sources of bias in sampling that should be avoided. Definition of common terms used in sampling. Sampling, the process of selecting a number of study units from a defined study population. Is a process of selecting a number of study units from a defined study population. For studies which involve only small numbers of people, all of them can be included in the investigation. What you say, all of them can be included into the investigation, which means all of them may constitute a sample. That's why you say all of them may be included to investigation. That is actually the meaning. So for research which focuses on a larger population for practical reason, it is only possible to include some of its members in the investigation. It is important to draw a sample from the total population. What does it mean? Mfano, unataka kufanya a certain research, maybe Ilala district. Umeona? But Ilala district ina watu wengi ambao kufanya research ukakumweka kila moja kuwa sample ya research inakuwa ni hectic na itachukua muda mrefu na you might end up in confusion. So you choose some of the sample. It depends na your study design. If it's about the primary school student, you know all schools in Lala, so you will select some schools, few of them, because you cannot do all of them. And if it's student, maybe class 4 student, you know there are so many schools with class 4. So what you'll actually do, you'll do sampling, selecting some from the school that you selected to run your research. So for research which focuses on a larger population for practical reason, it is only possible to include some of its members in the investigation. It is important to draw a sample from the total population. So population ile ni kubwa, lakini unaweza ukawak na wachache ambao ni enough kuleta result ambayo itapresent wengi. So tunasema hicho kitendo mtu kafanya sampling, that you have done sampling there. So let's see the sampling unit. The unit of selection in the sampling process: person, a school, a household, etc., etc. This is what I was trying even to explain up there. The unit of selection in the sampling process: example, person, a school, a household, etc., etc. But we also have a sampling frame, a list of units from which a sample is to be picked. Sample frame is a list of units from which a sample is to be picked. As we said, maybe we want to draw a sample from primary schools, class 4 student, Ilala district or Temeke district. It's from there you cannot work with all. You learn like the schools in Temeke district. Maybe there are 25 schools. You may opt to work on 5 schools. Are you getting? So these 25 schools is what we call sampling frame. But the sampling units are inakuwa ni hizi chache sasa ulizozichagua toka kwenye 25. So we have sampling fraction. Sampling fraction or sampling ratio is a proportion of sampling units to be picked from a specified sampling frame. This sampling fraction ni unachukua the number of units. For example, maybe out of 25, umechagua 12 schools or 10 schools. So to get the sampling fraction is 10 over 25. Are we getting the point? How this is clear. Then we have sampling interval, an interval at which units are picked from sampling frame when systematic sampling is done. This we're going to see when this systematic sampling, what it's all about. And we're going to see the sampling interval. But now, you should know an interval at which units are picked from sampling frame when the systematic sampling is done. But when we say sampling frame, it's the whole population. And the sampling unit is that sample you pick from a sampling frame for your study. Identifying the population to be studied. Study population. Collective of study unit for which the value of the varieties of interest could possibly be determined. Identify the populations to be studied. We must consider the following question. Identifying the population to be studied, we must consider the following question. What is the group of people, study population, we are interested in from which you want to draw a sample? For example, you may have a population tongue ruler and non-tongue ruler. Are we getting the concept? Maybe among level 6 college student, Kigambon district. Then what is the group of people? You have defined the group of people, and also you defined the variable you're interested in from which you want to draw a sample. How many people do we need in our sample? How will these people be selected? You know, you have defined the characteristics of the study or the study design. Now, how many people do we need in our sample? Or how will these people be selected? And the sample frame is there. How do you select these people from that population, maybe without bias, according to your research design? So the study population has to be clearly defined. For example, according to age, sex, residence. Otherwise, we cannot do the sampling. Each study population consists of study units. The way we define our study population and our study unit depend on the problem we want to investigate and on the objective of the study. As I said before, the study design and the objective of the study, and actually what you want to investigate. Representativeness. If researchers want to draw conclusions which are valid for the whole study population, which require quantitative study design, they should take care to draw a sample in such a way that it is a representative of that population. Okay, refer what I've said before, when you have this sampling frame, you already have a sampling frame. And these people you pick from this sampling frame and call them sample or sampling unit or sample unit. It means that these are representing the ones who remains. That's why now we are trying to elaborate things here. We said if researcher want to draw conclusions which are valid for the whole study population, which requires a quantitative study design, they should take care to draw a sample in such a way that it is representative of the population. A representative sample has all the important characteristics of the population from which it is drawn. This is an example. This is the whole study population. Then this is the part you took. So all this is the sample frame. And this is the sample unit, which has been drawn from the sample frame. Are we getting? So we call it what? Sample unit. So when you remove them from the study population, the process we call it sampling, as we have seen the definition of sampling. Let's see the common method of sampling. The common method of sampling are simple random sampling, stratified sampling, cluster sampling, systematic sampling, convenience sampling. Let's see the random sampling or probability sampling for quantitative data. If the aim of the study is to measure variables distributed in a population, example diseases, or to test hypotheses about which factors are contributing significantly to a certain problem, then it is important that the findings obtained from a sample to represent the total study population. It is important that the findings obtained from the sample to represent the total study population. This will say it's random sampling or probability sampling for quantitative data. Let's see probability sampling. We say probability sampling involves using random selection procedures to ensure that each unit of the sample is chosen on the basis of chance. All units of the study population should have an equal or at least known chance of being included in the sample. It requires listing of all study units exist or can be compiled. This listing is called sampling frame. Types of random sampling. Types of random sampling. As we say, we have cluster sampling, multistage sampling, simple random sampling, systematic sampling, and stratified sampling. Simple random sampling. The simplest form of probability sampling. Simple random sampling is the simplest form of probability sampling. To select a simple random sample, you need to make a search for an existing numbered list of the units in the population from which you want to draw a sample. Decide on the size of the sample. Select cluster sampling, the required number of sampling units using a lottery method. For example, when I say lottery method, for example, a simple random sample of 50 students is to be selected from a school of 250 students. Using a list of all 250 students, each student is given a number. So 1 to 250. And this number are written on small pieces of paper. All the 250 papers are put into the box, after which the box is shaken vigorously to ensure randomization. Then 50 papers are taken out of the box and the numbers are recorded. The student belonging to these numbers will constitute the sample. The student are 250, and from there you need 50 students from this start population of 250 students. You cannot just say Rhoda come, Doro come. No, it doesn't go that way. For you to avoid this biasness, then utaandika namba 1 mpaka 250, then maybe utaziweka mahali alafu unazichanganya zile namba, then you'll allow these 250 students to go and pick their numbers. Kila moja atapick. So waliopata hiyo namba 1 hadi 50 ndo wanakuwa your sample. So no one at first is assured to be part of the research, but after this random sampling, the probability of anyone to be there, they have equal chance of being involved in the study. I think we understand this. But we see this picked 1 to 50 are the ones who will be there, and no complaint. Let's see systematic sampling. Individual chosen at a regular interval. For example, every 5th. Individual are chosen at a regular interval, for example, every 5th from the sampling frame. Ideally, a number to tell where to start selecting individual from the list is randomly selected. In this systematic sampling, maybe you have 1 to 250. Are we getting? Then you may start anywhere. For example, maybe starting from 7, then you pick number 7, Then from every fifth, when you reach 12, you pick another. When you reach 17, you pick another. Are we getting it? So in every fifth, you pick. Every fifth, you pick. If you start up maybe from 7, you count 8, 9, 10, 11, 12, 13, you pick to your sample. For hivyo, you pick your stuff. After every fifth, it's a sample. Someone is picked to be involved in the study. So this we call a systematic sampling. Let's go to stratified sampling. If it is important that the sample include a representative stats, unit of small group with specific characteristics, then sampling frame must be divided into groups or strata according to these characteristics. If it is important that the sample includes a representative stat, units of small groups with specific characteristics, then sampling frame must be divided into groups or strata according to these characteristics. For example, resident from urban and rural areas, or different religious or ethnic group, or random or systematic sample of predetermined size will have to be obtained from each group, and we call it a stratum. That is simple and common. I think anyone can understand this. Let's see the cluster sampling. Cluster sampling, the selection of group of study units, we call them cluster, instead of the selection of study unit individually. The selection of groups of study units in cluster instead of the selection of study units individually. Cluster often geographic units, example, districts, villages, organizational units, clinics, training groups, etc., etc. Multistage sampling. A multistage sampling procedure is carried out in phases, and it is usually involve more than one sampling method. Sampling method for qualitative data. Purposeful sampling strategies for qualitative studies. Extreme case sampling, maximum variation sampling, homogeneous sampling, critical case sampling, snowball or chain sampling. Yeah, we say sample size. We have something called sample size. Number of subjects selected to represent a given study population. Sample size in qualitative studies. There are no fixed rules for sample size in qualitative research. The size of the sample depends on what you try to find out and from what different informats or perspectives. If you want to explore how you can involve mothers in your health clinic catchment area, you try to find that out. For example, effectively in early detection and the treatment of pneumonia, you might decide to conduct some focus group discussions to assess mothers' knowledge, attitudes, and practices with respect to pneumonia. You could start with focus group discussions among lowly educated mothers and to among mothers with more education, who usually are of higher socioeconomic status. If research objectives is more complex, example, attitudes of males and females toward family planning, and has policy implication for a large area, your sample will be bigger. You might start with 4 focus group discussion, 2 among males and 2 among females, subdivided according to the socioeconomic status. In exploratory studies, the sample size is therefore estimated beforehand as precisely as possible, but not determined. Tips for determining sample size. The desirable sample size depends on the expected variation in the data of the most important variables. The more varied the data, the larger the sample size needed to attain the desirable level of accuracy. The more varied the data, the larger the sample size needed to attain the desired level of accuracy. Hii sentence inamaanisha kwamba unaweza ukawa na variable ambayo unaipima, si ndio? Labda unapima urefu, watu warefu na wafupi. Umeona, maybe unairelate na body mass index, you can see the desirable sample size depend on the expected variation in the data. You cannot find people ambao wanafanana urefu, wanafanana sana urefu, but actually ukijaribu kuweka into group interval, it's from there unaweza kupata labda wenye urefu wa sentimita 60 hadi ngapi, 102 hadi ngapi hadi ngapi. I think you understand this. So the more varied the data, the larger the sample size needed to attain the desirable level of accuracy. The desirable sample size also depends on the number of cells in the cross tabulations. A rough guideline is to have at least 5 to 10 study units per cell. For example, after conducting a focus group discussion and in-depth interviews in the study on attitude of men or women toward family planning, you might decide to conduct a bigger survey. If the exploratory study revealed that age and education appear to be important factor determining this focusing people use, compare P use in group with different level of education and of different ages. So, in this, if the exploratory study revealed that age and education appear to be important factors determine focus population use, or compare focus population use in group with different level of education of different ages. If you split each of these variables up in 3 categories and to select 4 categories of informats for male users, pause of female users, male non-users, or female non-users, you would have 12 cells in each table. In order to obtain 5 to 10 answers per cell, you will require 60 to 120 informants in each research area. I think I'll send the video, then you'll learn this method. Actually, what I'm trying to explain here, you get to see what we say cells, what does cells mean. So, don't worry, although I know that you understand the concept. So eventually sample size is usually a compromise between what is desirable and what is feasible. Now let's come to the reason for sampling. Why sampling? Sampling is necessary when the study population is very big and the resources are not adequate to reach everyone in the population. To avoid or minimize bias, sampling can save time and money. A sample study is usually less expensive than a census study and produces a result at a relatively faster speed. Sampling may enable more accurate measurement for a sample study generally conducted by trained and experienced investigators. Sampling remains the only way when a population contains infinitely many members. Sampling remains the only choice when a test involves the extraction of the item at the start. Sampling usually enables to estimate the sampling errors and thus assist in obtaining information concerning some characteristics of the population. Let's see the sources of bias in sampling that should be avoided. Bias refer to systematic error in sampling procedures, which lead to distortion in the result of the study. Can be consequences of improper sampling procedures, which result in the sample not being representative of the study population. There are several possible sources of bias that may arise when sampling. The most well-known source is non-response. The bigger the non-response rate, the more necessary it becomes to take remedial action. The bigger the non-response rate, the more necessary it becomes to take remedial action. It is important in any study to mention the non-response rate and to honestly discuss whether and how the non-response might have influenced the result. Some biases in sampling. studying volunteers only. The fact that volunteers are motivated to participate in the study may mean that they are also different from the study population on the factors being studied. It is better to avoid using non-random selection procedures that introduce such an element of choice. Sampling of registered patients only. Patients reporting to a clinic are likely to differ systematically from people seeking alternative treatment. Missing cases of short duration. In studies of the prevalence of disease, cases of short duration are more likely to be missed. This may mean missing fatal cases, cases with short illness episodes, and mild cases. Seasonal bias. Problem understood, for example, malnutrition, exhibit different characteristics in different seasons of the year. For this reason, data should be collected on the prevalence and distribution of malnutrition in a community during all seasons, rather than just at one point in time. Tarmac bias. Study areas are often selected because they are easily accessible by car. Hii unakuta mtu anachagua sample kwa sababu ni mahali rahisi anaweza kwenda. Kwa anaangalia huku kuna lami, huku kuna njia ya nani? Ya udongo, kuna mashimo. Ngoja niende maeneo ambayo yana lami. Kwa hiyo inaitwa tarmac bias. Kwamba study areas are often selected because they are easily accessible by car. These areas are likely to be systematically different from more inaccessible areas. Let's see the ways to reduce the possibility of bias. Ways to reduce the possibility of bias. Data collection tools include a written introduction for the interviewer to use with potential respondents. Should be pre-tested. If necessary, adjustments should be made to ensure better cooperation. If non-response is due to absence of these subjects, follow-up of non-respondent may be considered. If non-response is due to refusal to cooperate, an extra separate study of non-respondent may be considered in order to identify to what extent they differ from respondents. Another strategy is to include additional people in the sample so that non-respondents who were absent during data collection can be replaced. Another strategy is to include additional people in the sample so that non-respondents who were absent during data collection can be replaced. This can only be justified if their absence was very unlikely to be related to the topic being studied. The bigger the non-response rate, the more necessary it becomes to take remedial action. It is important in any study to mention the non-response rate and to honestly discuss whether and how the non-response might have influenced the result. The key point to our study, sampling is the process of selecting a number of study units from a defined study population. Bias is sampling. In sampling is a systematic error in sampling procedures which leads to distortion in the result of the study. Evaluation, these are the questions. If you understood the content of the session, at least you'll be able to tackle these evaluation questions. What are the reasons for sampling? Kwa nini tunafanya sampling? What are the source of bias in sampling that should be avoided? The source of bias or bias in sampling and sampling that should be avoided. Thank you very much. Let's meet in our session, our lesson 14, and we see how to determine the sample size. Thank you for your time.